Okay, now we're going to move on to part 2 about cells. Okay, so organelles. Okay, organelles are like little organs. Okay, so the first organelle we're going to look at is a ribosome. Okay, a ribosome. Okay, it's going to be a small little ball of RNA and protein that makes proteins. Okay, so there are 2 types of ribosomes. Sorry, I was on the wrong thing. So ribosomes are like this little circle, and it basically consists of proteins and ribosomal RNA. Okay, and it's the site of protein synthesis where proteins are actually made. Okay, and like the way I like to think of this is slightly different than both. Okay, I like to think of the ribosomes as little individual factory workers. Okay, that make proteins. Okay, so proteins are the end product. The ribosomes are the things that are making the proteins. There are 2 types of ribosomes. Free ribosomes, which float around inside the cytosol. The job of free ribosomes is to make proteins that the cell itself needs, okay, inside the cell. And then there are bound ribosomes, which are ribosomes that are attached to the ER. Okay, these ribosomes will make proteins that either go to the plasma membrane or beyond, so outside the cell. Okay. Okay. The endoplasmic reticulum. Okay, endoplasmic, inside the cytoplasm. Reticulum is a series of membrane-walled envelopes and tubes. Okay, so reticulum. Okay, so there are 2 types of ER. Okay, ER can be either rough or smooth. So when we first invented microscopes, the microscopes kind of sucked. Okay, and so when we use that to look at ER, okay, what we saw was 2 types of ER. ER that was rough and ER that was smooth. Okay, ER that was rough, the reason it was rough is because it has ribosomes attached to it, and those were attached to protein making. Okay, the smooth ER, okay, it's not going to have ribosomes, so it's not going to be involved in protein making. It's going to be involved in other things. Okay, making sugar, making fats. Okay. Okay, so you can see that here. You can see this portion is rough. Okay, and you can see the little dots here, and you can see here that's much smoother and slightly smaller in terms of how big the little tubes are. Okay, smooth ER, rough ER. Okay. Okay, so rough ER, okay, ultimately makes proteins that will be exported, okay, either to the cell surface or outside the cell. Okay, so the next place that you can need to go is something called the Golgi apparatus. The Golgi apparatus functions in a lot of ways like a post office. Okay, if you think about what goes on in the post office, you receive a bunch of things out of the post office, you collect everything there, you tag it, you label it, you package it, and you ship it. That is what occurs at the Golgi apparatus. Okay, you receive things from the rough ER, you package it and label it, okay, until you ship it out. Okay, and so here you can see the whole thing, rough ER, Golgi apparatus outside. And so when you look at a cell, okay, sometimes they don't do a drawing like this, but if you actually look at a cell, it's always gonna be like this. So you always see rough ER, Golgi apparatus, outside. Okay, the Golgi apparatus is always gonna look like a stack of pancakes. Okay. Okay. Lysosomes. Lysosomes get pinched off the rough ER or the Golgi apparatus. what lysosomes do, lysosomes have digestive enzymes. They are the cell demolition group. So we'll explain this a little bit better and call them the recycling center for the cell. Okay, so when a cell uses a lot of product, uses a lot of proteins and stuff to get through the day. And as you use them, okay, those products become slowly damaged, like anything else you guys use, right, in your home. Those things slightly get damaged, slightly every time you use them. And over time, that damage builds up. Okay, and eventually, you have to get rid of it. Okay, the lysosome is where you get rid of it. You send it there, and it's a recycling center, and it breaks everything down into individual components again, and then you can reassemble using the original components. Okay, if there's a disease where your lysosomes aren't working properly, it's called Tay-Sachs, and it's a horrible disease. Okay, and affects children, babies actually, like, so if you get Tay-Sachs, you're dead by about 4. You start seeing the major symptoms by about 2, and you're dead by about 4. Okay. Okay, mitochondria. I'm almost certain all of you guys know what mitochondria are. And almost all of you guys, if I ask you guys what mitochondria is, you will say almost ubiquitously the mitochondria is the powerhouse of the cell, and that's like in every high school biology textbook ever, and it's more or less true. Okay, the mitochondria generates most of the cell's energy. Okay, it is a really complex organelle. Okay, what it actually is actually a bacteria that has evolved to live inside your cells. Okay, this is why they have DNA inside the mitochondria. Okay, that DNA is maternally inherited, so you get all your mitochondria from your mother. So that mitochondrial DNA is inherited from your mother, and you can use that to trace lineages. Okay, so for example, you can trace all of humanity back to a single maternal ancestor that lived about 300,000 years ago. Okay, you can also use it to trace more recent maternal lineages. So, and this is maternal, mother to daughter, not so mother to son, but son doesn't go on to the next generation. Okay, so one of the coolest examples of this is Richard III. Okay, so I love history, so you'll see a lot of history related things. Richard III was a king of England who died on a battlefield, and they, really hastily buried him somewhere. And that location was, we roughly knew where we buried him, but not really. And about 5 years ago, people were digging up an old church parking lot. Okay, and they found the skeleton. The skeleton was deformed like Richard III was, and it had a big hole in the head where he was allegedly hit by axe. So they were relatively certain that this was Richard III, but they weren't sure. So what they did was they take, extracts of his DNA, maternal mitochondrial DNA, and compared it to his mother's daughters to the present day women who were related, and they compared those extracts and they matched. So they were sure that this was Richard III. Okay. So here is mitochondria. Mitochondria always has a little loop of DNA in there. Okay. Okay. Peroxisomes. Okay. Peroxisomes are the toxic waste removal system of the cell. This one, the textbook has pretty much a better, really good explanation. So we breathe oxygen every day. Okay, oxygen is actually slightly toxic to us, and therefore we can't leave oxygen by itself, really. Okay, oxygen by itself tends to form things called free radicals. Okay, and the free radicals cause damage, and poisons also cause free radicals, which cause damage. Okay, the peroxisomes break down the free radicals. Okay, and that's why they're called oxidase enzymes. Okay, they undo the damage caused by oxygen. Okay. Okay. The cytoskeleton. Okay. The cytoskeleton is an elaborate network of rods. Okay. There are microtubules, microfilaments, intermediate filaments. Okay. You'll see I'll post a video for you guys to watch. Okay. Watch that video. You'll see all these cytoskeleton filaments. You'll see what they actually do. Okay, this is actin. Okay, this is intermediate filaments. Okay, and then this is microtubules. The most important one of these three are the microtubules. Okay, you'll see that the actin is everywhere, and the intermediate filaments are basically everywhere and scattered evenly. The microtubules are not. Okay, if you guys look at this very carefully, you'll see there's a center spot where all the microtubules actually go to and come from. Okay, that is something called the centrosome. Okay, centrosome, something near the center, and you see all the microtubules go there. This is the freeway system of the cell. Okay, later on in the video, they'll show you a cell walking, protein like walking, carrying a big sack of stuff. That is a microtubule that is worth walking on. Okay, so microtubules, in addition to providing support for the cell, actually provides a freeway system for the cell. Okay. Okay. In the middle are centrosomes and centrioles. Okay. This one we're going to skip for now. We'll come back to centrioles and centrosomes. Okay. A centrosome is basically something that consists of two centrioles. Okay. All right. We'll skip inclusions. Okay. The nucleus, okay, is the control center of the cell. The DNA directs the cell's activities. Okay. It provides instructions for protein synthesis. Okay, so here is the nucleus. Okay, the nucleus is where your DNA is kept. I'm sure most of you guys already know that one too. The nucleus is where your DNA is kept, and because your DNA is important, it is important that the nucleus is more protected than the rest of the cell. Okay, so to protect it more from the rest of the cell, the obvious thing is wrap another membrane around it. Okay, this membrane is called the nuclear membrane or the nuclear envelope. Okay, it's somewhat similar to the plasma membrane, but tighter. Okay, so less things can come in and out of it. In fact, the only things that can come in and out of it, okay, have to go through these little holes. The things that look like little flowers on there, they're actually little holes called nuclear pores. The nuclear pores decides what can enter and what can exit the nucleus. Okay, so the only way to get in and out of that nuclear envelope is to go through the nuclear pore. Okay, in the middle of the cell, of the nucleus, is another little tiny structure called the nucleolus. It literally means little nucleus, but that inside thing is called nucleolus. The nucleolus, okay, is where ribosomes are actually made. Okay. Okay. We'll end this here today. I'll pick this part up tomorrow when we talk about cell division and the cell cycle. All right.